Okay, part two, where we left off at taxonomy must change with evolutionary changes and new knowledge. It is far more important to have a taxonomic system that reflects our current knowledge than have a system that never changes. Creating a taxonomic system that provides an organized overview of all living things and how they are related to each other poses certain problems. Two such problems arise at opposite ends of the taxonomic hierarchy. One, deciding what constitutes a species, and deciding what constitutes a kingdom, or in which domain a kingdom belongs. In the first case, taxonomists try to decide how much diversity can be tolerated within the unity of a species. In the second, taxonomists try to decide how to sort the diverse characteristics of living things into categories that reflect fundamental differences of evolutionary significance. In most advanced organisms, such as plants and animals, species that reproduce sexually are distinguished primarily by their reproductive capabilities. A male and a female of the same species are capable of DNA transfer through mating and producing fertile offspring, whereas members of different species ordinarily either cannot mate successfully or will have sterile offspring. Morphology, structural characteristics, and geographic distribution are considered in defining species. So remember morphology, the structural characteristics. In bacteria, such criteria normally cannot be used in defining a species, primarily because lateral gene transfer, genetic recombination. Lateral gene transfer is genetic recombination. Among bacteria has been very common in evolution, but morphological differences are minor. A bacterial species is defined by the similarities found among its members. Properties such as biochemical reactions, chemical compositions, cellular structures, genetic characteristics, and immunology features are used in defining a bacterial species. So this is really important. I'm going to read it again. Properties such as biochemical reactions, chemical compositions, cellular structures, genetic characteristics, and immunological features are used in defining a bacterial species. Identifying a species and determining its limits present the most challenging aspects of biological classification for any type of organism. Before taxonomists turned their attention to microorganisms, the two-kingdom system of plants and animals worked reasonably well. Anyone can tell plants from animals. For example, trees from dogs. Plants make their own food, but cannot move. And animals move, but cannot make their own food. Simple enough, or is it? In this scheme, how do you classify Euglena, a mobile microorganism that makes its own food? How would you classify jellyfishes and sponges, which are modal or immodal depending on their stages of life? And how do you classify colorless fungi that neither move nor make their own food? Finally, how do you classify slime molds, organisms that can be unicellular or multicellular and mobile or immobile? Obviously, many organisms pose a number of problems when one tries to use a two-kingdom system. Developments since Linnaeus's time. Remember Linnaeus, The problem of classifying organisms was first addressed by the German biologist Ernst H. Haeckel, capital Haeckel, in 1866. So remember Linnaeus and then Haeckel, when he created a third kingdom, the Protista. He included among the protists all simple forms of life such as bacteria, many algae, protozoa, and multicellular fungi and sponges. Haeckel's original term, Protista, is still used in taxonomic schemes today, but it is now limited mainly to unicellular eukaryotic organisms. Classification of bacteria has posed taxonomic problems over the centuries and still does. Until recently, many taxonomists regarded bacteria as small plants that lacked chlorophyll. As late as 1957, the 7th edition of Bergey's Bergey' Manual of Determinative Bacteriology, a work devoted to the identification of bacteria, considered bacteria to be unicellular plants. Changes in this viewpoint came as the tool to study bacteria were developed. First, light microscopy and staining techniques were used to describe the structure of cells. Second, electron microscopy was used to study the ultrastructure of cells and composition and chemical reaction in cells. One of the most important discoveries from these various studies was that DNA looked and behaved differently during cell division in bacteria than in cells whose DNA is organized into chromosomes with the nucleus. Hold on, that was pretty disgusting, but I'm a human being. I don't know why people just don't drink seltzer water. They drink soda. They're idiots. Okay. Anyways. Where were we? Okay. Studies of structure and function of cells also led to the recognition of two general patterns of cellular organization: prokaryotic and eukaryotic, basing taxonomy on these two different patterns of cellular organization was proposed as early as 1937. Various taxonomists such as Copeland, Steiner, Van Niel, and Whitaker worked in the late 1950s. Okay, we always have to know names. Alright, so first we have Copeland and Steiner and Van Niel and Whitaker in the 1950s placed bacteria in a separate kingdom of anucleate, lacking a cell nucleus organisms rather than with organisms that have true nuclei. In 1962, Stanier, S-T-A-N-I-E-R, it could be Stanier, it could be French, and Van Niel stated, the distinctive property of bacteria is the prokaryotic nature of their cells. Now, what does that mean? The distinctive property of bacteria is the prokaryotic nature of their cells. So, a eukaryote can have whatever. I'm just going to read the book. In 1956, Lynn Margulis and H.F. Copeland proposed a scheme of classifying prokaryotes and eukaryotes by the following four-kingdom system of classification: Bonera, all prokaryotes, including true bacteria and blue algae; Protoctista. Protoctista. All eukaryotic algae, protozoa, and fungi. Plantae, all green plants. Animalia, all animals derived from a zygote, a cell formed by the union of an egg and sperm. These taxonomists who proposed that evolution from prokaryotic to eukaryotic life forms taken place by endosymbiosis. Remember the endosymbiotic theory that the mitochondria was its own little cell, and then it got phagocytized by the bigger cell, and it became part of it. That's the endosymbiotic theory from chapter 4. R.H. Whitaker. So far we have Stanier and Van Niel stated the distinctive property of bacteria is the prokaryotic nature of their cells. Then you have Margolis and Copeland proposed the scheme of classifying prokaryotes and eukaryotes by four-kingdom system. So Margolis and Copeland, the four-kingdom system of Monera, Protoctista, Plantae, and Animalia. Now, those taxonomists also propose that evolution from prokaryotic to eukaryotic forms had taken place by endosymbiosis. I talked about that. Now we have R.H. Whitaker, felt that endosymbiosis could not account for all the differences between prokaryotes and eukaryotes. He also felt that a taxonomic system should give more consideration to method organisms used to obtain nourishment, autotrophic nutrition by photosynthesis, and heterotrophic nutrition by the ingestion of substances from other organisms had been considered in earlier taxonomies. Absorption as a sole means of acquiring nutrients had been overlooked. To Whitaker, fungi which acquire nutrients solely by absorption were sufficiently different from plants to justify placing them in a different kingdom. Also, fungi have certain reproductive processes not shared with any other organisms. Consequently, Whitaker proposed a taxonomic system in 1969 that separated the Protoctista into two kingdoms, Protista and fungi, but retained the Monera, Plantae, and Animalia. Finally, through refinements of Whitaker's system by several taxonomists over the past few decades, the five-kingdom system was created. Okay, this is going to be part 3.